Okay, hello and welcome back. This is week 3 of the NPTEL course Experimental Biochemistry 2026, and this course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor Soumya from the Department of Bioscience and Biotechnology. I am Beas Mukherjee, a research scholar from the Department of Bioscience, Biotechnology and a Prime Minister's Research Fellow. And in this class, we are going to talk about different EM radiations, different spectroscopic techniques, Jablonski diagram, and we'll solve related questions, okay, based on this. So based on all these topics. So hopefully this class is going to be of some value to you. And let's begin with the class. As the students join, we'll quickly brush upon the different topics that are there. Okay, so we'll start with EM radiations. Let's talk about EM radiations. Electromagnetic radiation is defined as the propagation of energy through vacuum or any medium at the speed of light. It is made up of electric and magnetic field vectors oscillating at right angles to one another. So according to the quantum theory, the electromagnetic radiation is a flow of photons, also called the light quanta, which is the plural for quantum, through space. Now photons are packets of energy that always move at the universal speed of light. Now we know that light has the property of both radiation as well as particle. Right? It has both particle nature and radiation nature. And when we are talking about EM radiations, we are looking at the wave nature of light. But then when we are talking about photons, we are talking about the particle nature of light. Okay. Now, electromagnetic radiations include a vast spectrum of energy ranging from low energy radio waves to high energy gamma radiations. Some common forms of electromagnetic radiations include microwave, television waves, radio waves, infrared radiation, visible light, ultraviolet radiation, X-ray and gamma rays. So each form has different wavelengths and energy levels, resulting in unique properties and applications. Okay, now we'll move on. We'll look at the different forms of electromagnetic radiations. Each electromagnetic radiation has its specified range of wavelength, frequency, and hence its respective energy. Okay, so since the speed is same for all, which is the speed of light, different radiations will have different energy. Now we'll talk about those. Okay, so waves. Now what are wave? Wave is something that carries energy. Okay, so waves are disturbances that transfer energy from one place to another without the involvement of matter. So now waves can be of two different types. One is longitudinal wave and the other one is transverse waves. Okay, so what are longitudinal waves and what are transverse waves? Now longitudinal waves, in the longitudinal waves, the particles of the medium vibrate parallel to the direction of the wave propagation. Okay, producing alternating regions of compression and rarefaction. Sound waves traveling through air is a classic example. Now you can see in the figure that the direction of oscillation is parallel to the direction of the wave. Okay, and it is shown in the case of your longitudinal wave, which is shown in the left hand side figure. Now in the right hand side figure, we see a transverse wave, and from the previous figure, we saw that all these EM radiations that we have, they are all transverse waves. Okay, so here in the transverse wave, what happens? The particles vibrate perpendicular to the direction of wave motion, forming characteristic crests and troughs. So crests are these high points and troughs are these low points. Okay, and as seen in waves on a stretched string. So when we take a stretched string and we flick it, it forms these waves, right? And also on the and when we throw a stone in the pond, we will see these ripples on the surface of water. So all of these represent longitudinal all of these represent transverse waves. Now, while longitudinal waves require a material medium to propagate, the transverse wave can exist both in a medium and, in case of electromagnetic waves like light, even in vacuum. Okay. Now we'll talk a little bit about the electromagnetic radiations. So electromagnetic radiations are generated when electrically charged particles such as electrons move through a medium or empty space. So these particles carry energy as they travel, creating a wave-like pattern of electric and magnetic fields that we can see in this figure. Okay, so electromagnetic radiations travel as transverse waves. The oscillations of the electric and magnetic field vectors are perpendicular to the direction of propagation of the wave and perpendicular to one another. Okay. Now wavelength represents the distance between two adjacent points in the same phase. Okay. And frequency gives the number of wave cycles per second. So that is your frequency. Amplitude refers to the maximum displacement of a wave from its equilibrium position. So that is the amplitude. So all of these different properties of a wave are shown in the figure for your understanding. Okay. Now all forms of electromagnetic radiations travel in vacuum at the speed of light, which is 2.9 into 10 to the power 8 meter per second. And that is often rounded off to 3.8 meter per second. Okay. 3 into 10 to the power 8 meters per second. Okay, now here is the entire electromagnetic spectrum for our understanding. So I would recommend you that you remember the range of the wavelength for different types of electromagnetic radiations. Okay, so for example, let's say that, or you can also remember the frequency. So if you remember any one, that would be okay, because the speed of light is constant, and that is how you can actually calculate and find the wavelength. If you know the frequency, you can find the frequency if you know the wavelength. Okay, so for gamma radiations, you see the particular range of frequency and wavelength is given. Then after that comes the X rays, then comes the UV light, and just right after that comes the visible light containing VIBGYOR, which is violet, indigo, blue, green, yellow, orange and red, that we can see, the colors of the EM spectra that we can see. Then comes the infrared radiation. So infrared radiations are responsible for producing heat. So all the devices that we have, all the electrical appliances in which some kind of heating is done, most of these use these IR radiations. Okay. Then the radar waves and microwaves. Then comes television waves and radio waves. So these are the different electromagnetic radiations, and the entire spectrum is given to you. Okay. So here the specific wavelength, frequency and energy for different electromagnetic radiations are given for your understanding. The electromagnetic spectrum is a range of frequencies, wavelength, photon energies with frequencies from below 1 Hertz to above 10 to the sorry, 1 second. Please give me a second. Okay. Okay, hello. So we are talking about electromagnetic radiations in today's class. Okay, and let's continue. So we are looking at the different electromagnetic radiations that are there. So electromagnetic radiations is a range of frequencies, wavelengths and photon energies with frequencies from below 1 Hertz to above 10 to the power 25 Hertz. Okay, so corresponding to all electromagnetic radiations is in the frequency order of frequency, in the increasing order of frequency and decreasing order of wavelength, that is given in this figure. Now we see that for the highest wavelength, it is radio waves, and therefore correspondingly these have the least frequency. then comes the microwaves, then comes the infrared radiations, then optical range, that is the light that we can see. Okay, so VIBGYOR comes in this range. Then we have UV light, we have X-ray, and finally we have gamma radiations. So you are recommended to remember at least the wavelengths or the frequency, okay, any one of these two, because you already know the speed of light, so you can always calculate and find out the other one. All right, so now we look at what's next. Now, since we know about all of these different electromagnetic radiations, we would now we would get to know about the uses of these electromagnetic radiations. Okay, so for radio waves, we see that broadcasting and communications are used, and microwaves are important for cooking, radar communication, satellite communication. Infrared radiations is important for thermal imaging and remote controls. Okay, and visible light is important for human vision, optical communication, and UV light is important for sterilization, UV lamps, forensics, etc. So you also take a look at the wavelength, range of the different wavelengths here. So X-ray is important for medical imaging, security screening, etc. Gamma radiations are used in medical imaging, cancer treatment, nuclear medicine, etc. Okay, so the three figures that are given, the first one shows IR, which is infrared radiation. This is getting emitted from a star, because of which we are seeing that it is red in color. IR is giving this red colored effect. Then is white light. There is a prism dispersion that we see, and the last one is X-ray used for medical imaging. Okay, right. So now we will talk about the applications of these different radiations in a little more details, because these come as match the following questions in your GATE and CSIR NET, ICMR, DBT, all of these different exams. Okay, and also NPTEL examinations. Now, uses of gamma radiation. So though gamma radiations are biohazardous in nature, they can be controlled and used for various important purposes. It is used in the treatment of cancer without surgery. The cancerous tumor is subjected to gamma radiation, which kills its DNA. It is used to sterilize surgical instruments. It is also used in the food industry to kill harmful bacteria. Now, infrared radiation is also referred to as heat waves or thermal waves, as they have a heat inducing property. So, infrared rays are used to make infrared heaters that are used for therapeutic purposes requiring heat, often physiotherapy, pain relief during injuries. These are the different purposes for which infrared radiations are used. Now, okay, we have people joining. Okay, hello. So in this class, we are talking about EM radiations, and right now we are talking about the utilities of infrared radiations. Okay, so infrared radiation is also referred to as heat waves or thermal waves, and IR radiations are classified as near infrared and far infrared radiations. Now, what are these? So, near infrared radiations are used in electronic appliances such as TV remote sensors and photography. And far infrared radiations, they are more thermal. So, that means they provide more heat energy. Anything generating heat gives out far infrared radiation. Even the human body at 37 degrees centigrade, which is our normal body temperature, it gives off infrared radiations of around 800 nanometer wavelength. Okay, now we'll go for the next part. So you see that this near infrared, short wavelength infrared, medium infrared, long wavelength infrared, and far infrared. These are the different variations of infrared radiations and their specific applications and their ranges. Wavelength range is given to you, so you can have a look at this. I will not go through this now because we'll be wasting the time of the class, right? So it's best if you go through this. So if there are any questions regarding this, then you'll be able to answer. Okay, so we'll continue. Now we come to one important MCQ. Now this is an MCQ that you can try solving. Okay, so this this is a simple MCQ, week 0 MCQ. This can look discouraging at first because here you are supposed to remember the wavelength range of UV light. But it is not very difficult. If you give some thought to it, you will understand. All you need to do is remember the starting and ending of the visible light spectra. And you know that UV comes just next to visible light, right? So it is having wavelength just less than visible light. So then based on that, what do you think is the wavelength range of UV light? Now I can give you the hint that for visible light, the wavelength range is 400 nanometer to 700 nanometer, right? So now you understand that based on these options, which one would be UV light. You can give it a try. Okay, so you can just write in the chat box whatever you think is the right answer. You can answer in the chat box. Okay, try answering. It's very simple. So whichever you think is the right answer, you can just try to answer in the chat box. Okay. B. Okay, good. Great. Yes. Right. That is correct. B is the right answer. So if you look at this figure, you will see that for visible light, as I told you, 400 to 700 is the range. And the option that is given to you just below 400 is 10 to 400 nanometers. So this is how you can simply remember the ranges of these spectral like sections, okay, different waves. Okay, now we'll go to week 3 assignments. All questions of week 3 assignments. Okay, so we have one question at first. Which region in the electromagnetic spectrum has a wavelength of 0.01 to 10 nanometer? Now what comes before UV light? Tell me. That would be the right answer, right? So UV light, we know that this is 10 to 400. So whatever comes before UV light, whatever is even shorter than UV light when it comes to the wavelength, that would be your right answer here. So you can try answering whether it is X-ray, infrared region, ultraviolet region or radio wave region. Okay, so you are saying that it is A. Let's see. Yes, that is correct. X-ray is the right answer. Yes, so X-ray ranges from 0.01 nanometer to 10 nanometers. Okay, very good. Okay. Now we have this table once again. Gamma rays, the different frequencies, different electromagnetic radiations and the different range of wavelength is given. Okay, so you can go through this whenever you get time. Now, in different questions, they will ask you to find the energy of the EM radiations. So how do you find the energy? You already know the speed of light. You know the value of the Planck's constant, right? So there are only two equations. One is F is equal to C by lambda and the other equation is E is equal to hc by lambda, right? So based using these two equations, you can very easily calculate the energy of any particular EM radiation if you know its wavelength, right? Because speed is same for all. Now we try to answer this question. Okay, this is also a simple question. So whatever we talked about until now, just the last slide, if you keep that in mind, you will be able to answer this. Okay, so if this frequency of radiation is increased by 2 times, let's say that initially the frequency was F, now it has become 2F, let's say, right? So then what could be the change in the energy of the radiation? So you have to find out how is frequency related with energy. Try to recall these two equations that we talked about in the previous slide and you can answer the question. Okay. Try answering. B. Okay, good. Let's see. Yes, very correct. Right? So, just the two equations that you have to keep in mind and then you know that energy of the radiation varies proportionally with its frequency. So, if frequency becomes 2 times, then the energy would also become 2 times. Right? Okay. Now, we have another question. The wavelength of 4 electromagnetic radiations are given. Wavelengths are all given to you. Now they are asking you which is the highest, which of the following radiations have highest energy per photon. Also very simple. Try answering this. Okay. Which has the highest energy? Okay. So you are saying that it is C. Let's see. Yes, yes, that is correct. Very good. Yes, it is C. So what happened is in the previous question, it was frequency and energy, relationship between frequency and energy. And in this one, this is the relationship between energy and lambda, which is inverse. Right? So very good. Yes, good. Now we'll start talking. Now we'll start talking about spectroscopy. Okay, we'll try to understand spectroscopy. Now, spectroscopy is a very important topic, and you can expect this in every exam. Okay, so be it CSIR NET, be it GATE, be it TBT exam, be it this NPTEL examination, spectroscopy is something that has applications in many disciplines, and that is why this is a very important topic. So let's try to understand. It is very simple. Okay, so I'll try to explain as nicely as I can. Let's try to understand this. So interestingly, different substances can absorb and emit light at characteristic wavelengths. Okay, so this is the basic principle of spectroscopy. Let's say there is some substance. Let's say that it absorbs light at a particular wavelength and it emits light at a particular wavelength. Now the wavelength that we see also depends on which wavelength it is emitting. Right. So that is how this particular substance gets its color. Now, so see, spectroscopy is a technique used to study the interaction between matter, which is this substance, and electromagnetic radiation. So this involves analyzing the spectra of the emitted, absorbed or scattered light to gain information about the composition, structure and properties of the material. Okay. Now there are different kinds of spectroscopy. For example, there is atomic absorption spectroscopy, which deals with visible and UV light. Then there is, so basically we'll talk about this absorption spectroscopy today. There is infrared spectroscopy. So this involves, of course, only infrared radiations, and this is useful for identifying functional groups in organic molecules, etc. Then there is UV visible spectroscopy. We are going to talk about this. So here what happens? UV and visible light is absorbed here. So this is useful for quantitative analysis of different biomolecules. We are going to discuss this today. Then there is NMR spectroscopy. Now here there are very radio frequency range particles, okay, and proteins. They are also proteins and chemical compounds. They are often NMR active. Okay, so determining the molecular structure and identifying compounds. That is what NMR can be used for. Now it has a wide range of applications, and our lab actually specializes in NMR. Okay, so X-ray absorption spectroscopy. So this is again dealing with X radiations and studying electronic and structural information in solid liquid gases. Then there is Raman spectroscopy, which deals with visible and near infrared radiations. Okay, so we'll start talking about absorption spectroscopy first. So absorption spectroscopy is the simplest, but there is always one question from this Beer-Lambert's law. Okay, so you can consider this under UV visible spectroscopy also. So as we go, so this works on absorption spectroscopy. Okay, Beer-Lambert's law. Now let's try to understand this. So absorption spectroscopy is a biophysical technique which is used to measure the absorption of electromagnetic radiation by a substance as a function of its wavelength. So the basic principle of absorption spectroscopy is that includes UV visible spectroscopy. Okay, so involves passing light through a sample. So there is this sample through which light is passing, and measurement of the amount of light that is absorbed at different wavelength, that is done. So that is all about absorption. Then the extent of the absorption is related to the concentration of the absorbing species and the characteristic of the molecule's electronic structure. That is all about absorption spectroscopy, and this works on the principle of Beer-Lambert's law. So absorption spectrophotometer is the instrument using which this particular spectroscopic technique is studied. Okay, so this has a lot of applications. This is used in everyday research in the lab. Okay, so this has importance in quantitative analysis. So based on how much light is getting absorbed, you can calculate the concentration of the sample present. and qualitative analysis, so identifying functional groups and electronic transitions in the molecules, and monitoring reaction. So different reactions, whether they are progressing, whether they are not happening, the kinetics of the reaction, all of that can be studied using absorption spectroscopy. Okay, so this is a very simple technique but very powerful technique. Now we'll talk about this in a little more details. So this is your Beer-Lambert's law, from which you can expect one question every year. Okay, so what happens in Beer-Lambert's law? Beer-Lambert's law says that for a given material sample, the path length. So what is the path length? Let's say that this blue thing that you're seeing, this is the cuvette. So from the region from which light strikes and enters the cuvette to the region where it is leaving, that is called the path length. Path length is often taken as 1 centimeter. Okay, for 1 centimeter path length, and concentration of the sample are directly proportional to the absorbance of light. So if you know the absorbance of the sample, how much light is absorbed, if you know that, then you can calculate the concentration of the sample if the path length is known as 1. Okay, now there is another player in this equation, which is that is epsilon. So what is epsilon? Epsilon is actually the molar absorbance. Okay, so molar extension coefficient or molar absorptivity. So what is this? This is specific to the absorbing species. So let's say I'm taking a protein solution, and in that protein solution, let's say how many tryptophan residues are there, how many tyrosine residues are there, how many phenylalanine residues are there, that constitutes my molar absorption coefficient of that particular material. And if that is very high, then it will absorb a lot of light. If that is very less, then it will absorb very less light. Based on that, I will calculate the concentration of the sample. Okay, so here the drawback is that this particular material that I am taking, it should have this sufficient molar absorption coefficient so that we can calculate concentration from absorption. Okay, so if a particular protein, it does not have any aromatic residue, then you cannot calculate this concentration of that particular protein using Beer-Lambert's law and UV visible or absorption spectroscopy. Okay, so see, there are two formulae here. One is A is equal to epsilon into C into L, that we saw. The other one is A is equal to log base 10 I naught by I. So what is I naught? The light that is initially coming and striking. So that is the incident ray, that is I naught. And the light that is leaving after passing through the sample, that is called I. So I naught has more intensity compared to I, and the ratio of I naught by I, that is log of that particular ratio, gives you absorbance. Okay, so Beer's law states that the concentration and absorbance are directly proportional to one another, and Lambert's law tells that the absorbance and path length are directly proportional to one another. Okay, another thing is log of I naught by I is equal to epsilon into L into C, right? So that is the value for A or absorbance that we are substituting. Now we come to a question. So based on up till here, you've understood that Beer-Lambert's law is something very simple if you understand it. Okay, but then the questions that you can expect may not be very straightforward or direct. So let's look at this particular question here. It is not a very direct question, but then it's important that you practice it. Okay, so this is a gate level question for you. A compound X absorbs monochromatic radiation of wavelength 450 nanometer and follows Beer-Lambert's law. A solution of X shows 40% transmittance. So what is transmittance? We will look at it very quickly. Okay, so that means transmittance means how much is not absorbed, how much is transmitted. Okay, and the path length of the cuvette is 1 centimeter, which is supposed to be, right? And the concentration is 2 into 10 to the power minus 4 molar. So concentration is given to you, path length is given to you, and what else is given to you? The transmittance is given to you. So when the path length is doubled and the concentration is half, what will be the percentage of transmittance in the solution at the same wavelength? Okay, let's see. So even if you don't know the transmittance, very simply from here also you can actually calculate. So let's say that the path length is doubled and concentration is halved, right? So you know that all of these A is equal to C into L into epsilon. Okay, and you know that L is getting doubled, so that is 2L into C divided by 2. So both the 2's are getting cancelled, so it should not change. But when they are talking about transmittance, let's see, okay, how to solve this question. But normally absorbance is going to be the same, okay, technically. Now let's understand how this problem is solved. Okay, right. So let's see how this problem is solved. So see, A is equal to epsilon into C into L. Right. Now A is equal to minus log T. Now what is this T? Now we know that A is equal to log into I naught by I. Now if you are just making it reverse, then you will have to take it as minus T. Right. So instead of absorbance, we are talking about transmittance. So that is why we are taking T in this equation and minus log T, because it's exactly the opposite. Okay, now given transmittance is 40% or 0.4, and then like this they have done the calculation, exact thing that we said just now. So they and finally they see that the product Cl remains unchanged, right? So A is equal to minus log T, and we see that T2 is equal to 10 to the power minus A2, which is 10 to the power this minus 0.39, and from there we get that this is once again 40%. Right? So the question was very simple, but the way to solve it, if there are more questions on transmittance, then you can follow these equations and you can solve them. But for this question, you did not need so much of calculations. It was very simple. But then I gave you this question so that later on, if there are any more questions regarding transmittance, it will give you some hint, okay, on solving them. Okay, now we go to UV visible spectroscopy. Okay, and if you're not understanding, please stop me in the middle and tell me. Okay, I'll explain for you. Today we don't have too many students. Maybe you guys are having exams or something, but we have very low population today. That's okay. We'll continue. Okay. So UV visible spectroscopy. Now what's happening here? Until now we were talking about absorption of light, and that is being measured. So amount of light that is absorbed was getting equated with the concentration of the sample. Now we will talk about what light is emitted after some wavelength is absorbed. So whatever wavelength is absorbed, but some other wavelength gets emitted. Okay, and we will start talking about that. So see, UV visible spectroscopy involves measurement of the absorption of the UV light and visible light by molecules. So light sources, they are often tungsten lamp for visible light and deuterium lamp for UV radiation. Here what is happening? Ultraviolet light and visible light, they are getting absorbed. Okay, so monochromator. What is a monochromator? So if you look at the figure, you will see that from this tungsten lamp and D2 lamp, deuterium lamp, the light is getting emitted and there is a mirror and light is getting reflected. It is coming to the monochromator. Monochromator we have come across in our previous slides as well. You remember, I'm sure, that monochromator is actually selecting one specific wavelength only. Okay, and through that slit only one specific wavelength from this white light is going into our sample, and as it goes into the sample, you see that there is this beam splitter, okay, and mirror and reference. So there is a reference electrode, reference cell, and there is this sample, right? So that is the difference between absorption spectroscopy and UV visible spectroscopy. So here there is a reference. Then there is the detector, which is detecting the wavelength that is coming out from the sample as well as the reference, and then there is processing, and then we get the data. Okay, so the sample holder contains the sample in solution. Quartz cuvette ensures minimum absorption of UV light by glass and maximum by the sample, and detector measures the intensity of the transmitted light. Okay, so there is this quartz cuvette which is used because quartz absorbs very less light and ensures that more light goes to the sample. So based on whatever we have talked about until now, we will try to solve this question. Okay, so the correct path of a spectrofluorometer. So what is spectrofluorometer? This is the instrument used for UV visible spectroscopy. Right? So what would be the correct path? You can answer in the chat box. Okay, so first there has to be light from the source, then it is going to travel, and then finally there is going to be, okay, so you are saying that it is D. Let's see. Yes, correct. It is D. Okay, very good. Now we'll proceed. Acha. So now we come to something that is very interesting. This might be new for you. So let's try to understand this nicely. Alright. So now there is something called spectral shift. Let's say that this molecule, some kind of molecule you were having. Let's say you were having a biological molecule. Then you add some other molecule into it, because of which its spectral properties change. Right? So there is this spectral shift. Let's say it is absorbing more, absorbing at a more higher wavelength. That is called hyperchromic. So let's see what are the different types of spectral shift. Okay. So hyperchromic shift occurs when a molecule undergoes a change in conformation, interacts with other molecules, or experiences a change in its environment such that it leads to an increase in absorbance or fluorescence intensity. So the graph goes higher. Okay. 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 People are coming in. Let's admit them. Okay. So this is hyperchromic shift. Now there is something called hypochromic shift. What is happening? That means less intensity of absorption is there. Why is that happening? Let's see. Now hypochromic shift is observed when conditions lead to reduction in absorbance or fluorescence intensity. This can result from the changes in the molecule's own environment, such as decrease in solvent polarity or alterations in the molecular structure. Okay. Because of this, there is reduction in absorbance. Now there is something called hypsochromic shift. What is that? Hypsochromic is often associated with changes that results in higher energy transitions. For example, a shift in absorption or emission towards shorter wavelength might occur due to an increase in electron density or change in electronic structure of the molecule. So that means the shift is going towards the left. This is called blue shift. Okay, it's going towards the left. And there is something called bathochromic shift, which is the red shift. So that means the shift is going towards the right. So wavelength of light that is higher than the initial wavelength, that is getting absorbed more. Okay, so why does that happen? This shows indicate a decrease in energy levels. So this shift can occur due to different factors such as increase in solvent polarity, changes in the molecular structure, or interaction with other molecules. That is when red shift or bathochromic shift happens. Okay. Now all these shifts are given to you in the form of a table. So see, the different names are given. So parameter change, in case of bathochromic shift, this lambda max increases. That is the maximum wavelength of absorption. Okay, it is the central highest point in the curve. So that is red shift. For hypsochromic shift, it's coming, it's hypsochromic is actually blue shift. So lambda max decreases. Hyperchromic intensity increases, so epsilon increases, and hypochromic epsilon decreases, intensity decreases, keeping the concentration same. Right? Now, see, everything is given here. What it indicates about the molecule and the typical causes, those are also given. So for hyperchromic, you see that hyperchromic can happen when there is unfolding, dissociation, or increased conjugation, and this is seen in case of DNA denaturation and disruption of hydrogen bonding, etc. Hypochromic, that means it's hyperchromic, means it's basically more intensity of absorbance is there, and hypochromic, that is less intensity. Here DNA base stacking is happening. Now why is that happening? I'll tell you. Because these individual nucleotides, they are actually very potent in absorption. Okay, so they are the internal fluorophores inside the DNA, and when they are free, when the two strands are separate, they will absorb a lot more light. But when they are coming together, they are stacked. That is why they will absorb less light. That is why it is hypochromic when the DNA becomes double stranded, but it is hyperchromic when it is separating out. And then there is protein ligand binding, aggregation. These are hypochromic effects. Then we come to bathochromic shift. So solvent polarity increase and oxochromes are there. pH induced ionization. All of these different things are happening. And then there is blue shift. So protonation of the amino acids. We know about this. We have studied in the previous class. Okay. So amino acids are they are accepting H plus ion, that is called protonation, reduced conjugation and solvent effects, etc. All of these results in blue shift. Okay, so you can read about oxochromes. What are oxochromes? You can read. That is often an important question which is asked. Okay, so fluorophores, chromophores, oxochromes, all the three you must be knowing. In the upcoming slides, we'll talk about fluorophores and chromophores. Okay. Right. So now we'll try to solve this question. Okay. So based on your understanding until now, you should be able to solve this. So in this above graph, what kind of shift do you observe in B with respect to A? So A is there and then it shifts towards the left plus it goes up. So which two forms of shifts are involved here? Tell me, try to answer this. Hypsochromic, bathochromic, hyperchromic and hypochromic. These are the four names you can answer in the chat box. Okay. Oh, we already have the answer. Good. Okay. So you're saying that it is D. Let's see which is the right answer. Acha. So 1 and 3 are correct, it says. Why is that so? Because 1 is hypsochromic and 3 is hyperchromic. So yeah, we can see that it is hyperchromic, that is for sure. And which one was hypsochromic? So it is shifting towards the left, right? So if it is shifting towards the left, that means it is blue shift and blue shift is hypsochromic. So hypsochromic means that shifting to a shorter wavelength plus the intensity is increasing in B. That is why it is both hypsochromic and hyperchromic. So B would be the right answer. Okay. Now we'll talk about absorption versus emission spectroscopy. So now I was telling you that until now we were talking about absorption. Right, so now we are going to talk about emission spectroscopy. So what is that? Fluorescence spectroscopy works on the principle of emission spectroscopy. So emission spectroscopy means that some light is absorbed, but fluorescent light is often emitted by that sample molecule. Okay. So in emission spectroscopy produced when atoms are releasing energy, comprises of colored lines in the spectrum. So the colors that we see, they are often corresponding to fluorescence spectroscopy. It is helpful in figuring out the composition of a certain matter, and the type of photons emitted are helpful in figuring out what kind of element the substance is made up of. So we'll talk about this in details now. Okay, so now this is something called Jablonski diagram, which you need to understand very well for understanding fluorescence spectroscopy. So if you look at the diagram, you see that initially there is this ground state, okay, and then the electrons, they are absorbing light, they are getting transferred, they are actually getting excited and going to higher energy levels. Now there are different higher energy levels, for example S1 level and S2 level. In the respective energy levels also there are different vibrational states. What does that mean? Mean that for each energy level, it is not just one particular energy state, but multiple similar energy states and with very slight modifications at a particular energy level. And these are called different vibrational states. Okay. So now on absorption of the light, these electrons can go to different energy states. And then there is something called internal conversion. So it is traveling from the higher energy state to relatively lower energy state and giving out some energy. And then it is coming back to its ground state. While coming back to its ground state, it is again emitting light, and that light is corresponding to fluorescence. Okay. There is something called intersystem crossing, and fluorescence is responsible for, so singlet state of electron is responsible for fluorescence, but on intersystem crossing there is a triplet state form, and this results in phosphorescence. So what is the difference between fluorescence and phosphorescence? We are going to talk about this. Okay. So now these are all very natural biological phenomena. So you will see that in a lot of deep water fish and these deep water creatures, they have these bioluminescence properties. Okay, so often these are phosphorescent. Now see what is happening. So the Jablonski diagram is a graphical representation in the field of spectroscopy to illustrate the electronic transitions that occur in a molecule when it absorbs or emits photons of light. Now we will talk about all of these in details, okay, in the upcoming slides. So we'll start with the Jablonski diagram. This provides a visual representation of the energy levels and transitions between the electronic states within a molecule. Okay. Now see, the key features are electronic states. It depicts different electronic states of a molecule such as the ground state or the lowest energy state and various excited states or higher energy states. The transitions, the arrows that are going upward and downward, these are all arrows across the diagram represent possible transitions between the electronic states. These transitions are associated with the absorption or emission of photons. So energy level, the vertical axis represents the energy level of the electronic states. The higher energy levels positioned higher on the diagram. Okay, so energy axis goes this way. The y-axis is your energy axis. Now non-radiative transition. What are those? Some transitions are non-radiative. So that means that the energy is transferred within the molecule without the emission of light. These transitions are often represented by wavy lines. So we saw those intersystem crossing and we saw these internal crossing. Those are the wavy lines that are showing that within the molecule, some of the energy is lost as the electron travels. Okay, so it is not emitted, but it is just lost. Then there is radiative transition. So other transitions are radiative. That means involving either emission or absorption of photons as it is going up and it is coming down. So these transitions are represented by straight arrows. Okay. Now we have something more about Jablonski diagrams, fluorescence and phosphorescence. So in the context of the Jablonski diagram, there is something called internal conversion and something called intersystem crossing. So what are these? These are non-radiative transitions that describes ways in which a molecule can return from an excited state to the lower energy state without emitting a photon. Okay, so what is happening in case of internal conversion? In internal conversion, you see that internal conversion is basically there is this redistribution of electronic energy among the different vibrational and rotational modes of the molecule, ultimately leading to the transition to a lower electronic state without emission of light. So that is how internal conversion is happening. Then there is intersystem crossing. What is that? So in intersystem crossing, this is also a non-radiative process which involves transition between different spin states. For example, singlet to triplet or vice versa. What is that? We'll talk about this. Okay. So this process occurs when the molecule changes its electronic spin state without emitting a photon. For example, a single may transition to a triplet. Now, what is singlet and what is triplet? We look at this. So see, so singlet and triplet are terms used to describe different spin configurations of electrons within a molecule's electronic state. What is a spin? Now electrons can have upward spin, they can have downward spin, right? So property spin multiplicity. So okay, so we will not go to something that is very difficult to understand. We try to understand it in a simple way, right? So we know that one electron, it can have an upward spin, it can have a downward spin. Okay, so the spin multiplicity is related to the number of possible orientation of electronic spin. In a singlet state, all electron spins are paired. That means resulting in zero total spin. So if one is like this, the other one is like that. So the net spin, total spin would be zero, right? But so for example, if there is a transition between pi to pi star, so pi is bonding electron orbital and this is the anti-bonding, right? So there is no change in spin. So initially it was like this and it is going, one of them are going to the higher one. So the higher one would, higher electron orbital, higher energy state, and it would be like this, right? So there is no change in spin. But when it is going to pi to pi star, there is no change in spin, and then finally when it is reorienting itself, there is a change in spin. So it was initially like this, but when it is going here, this one is changing its spin to this, and that is when triplet state comes. So in a triplet state, two electrons have parallel spins resulting in a total spin of 1. So both of them are having downward spin or both of them are having upward spin. So singlet state is seen in case of fluorescence, whereas triplet state is seen in case of phosphorescence. Okay, in singlet state you will see that all the electrons are paired, and in triplet state two electrons have parallel spin, one has opposite spin. And typical higher energy, and this is typically lower energy, triplet is lower energy, and singlet is shorter, nanoseconds to microseconds lifetime, and triplet, because it is more stable, it is having longer lifetime. Okay, now we will try to solve questions on this. Now the questions wouldn't be very difficult to solve. I just explained this to you because you might have a lot of doubts about these spin states and all, and about fluorescence and phosphorescence. But let's try to understand the Jablonski diagram properly first, because there would be questions related to this. Okay, so this is once again a gate level PYQ, previous year's question for you. Try to solve this. So based on whatever we talked about the Jablonski diagram, this question is based on that. Okay, so try to understand this. Okay, you can answer in the chat box. So fluorescence, phosphorescence, internal conversion, and intersystem crossing. Right? So spin forbidden transition. Red, acha. Maybe A or C. Acha, acha. Good, good. Okay. Anyone else wants to give a try? Radiation-less transition between states of same spin multiplicity. Emission from excited singlet to ground singlet state. This should be simple. Excited to ground singlet. Right? Okay. And acha, so that's why you're saying it is A or C. Okay. Emission of long lifetime after removal of one excitation source. Let's see. Okay, let's see which is the right answer. Right answer is A. Now, why is that so? Because see, fluorescence means emission from excited singlet. Fluorescence we know is excited emission from excited singlet state to ground singlet state. Correct. Now, phosphorescence occurs from triplet state, has a longer lifetime and persists after radiate excitation is removed, and that is why this would be 4. Q would be 4 because of that. Right. Then there is internal conversion. Internal conversion is once again non-radiative, so it is radiation-less transition between the same spin multiplicity. If it is same spin multiplicity, then it is not phosphorescent, right? And intersystem crossing involves crossing of the state. So internal conversion is between same spin multiplicity, and intersystem crossing is spin forbidden transition between singlet and triplet states. So if you look at the diagram once again, you will understand this better. So intersystem crossing means that there is this crossing happening, but when it is internal conversion, it is same spin state, within that only the transition is happening. Right? So hopefully now you will remember this. Okay, we'll go to the next part, which is again fluorescence spectroscopy. Let's try to understand this. So fluorescence occurs when a molecule absorbs light at a specific wavelength, which is called excitation wavelength, and undergoes a brief electronic transition to a higher energy state. Then it returns to the ground state by emitting light at a shorter wavelength. Remember this, okay? Shorter wavelength. And the emitted light is called fluorescence. Its intensity and wavelength can be measured to gather information about the sample. Okay, so shorter wavelength means higher frequency of the wavelength, right? So higher frequency means higher energy. Now fluorophore. So fluorescent molecules are capable of absorbing and emitting light of longer wavelength. 1 second. Oh, sorry. It is longer wavelength, not shorter wavelength. because if it is longer wavelength, only then it will be of a relatively lower frequency. That means it is going to release some energy in the process, and that energy is released in the form of internal conversion. Right? Now it will make sense. Okay, let's continue. So tyrosine, tryptophan and phenylalanine, all of these are amino acids and these are natural fluorophores. Okay, they are natural fluorophores plus natural chromophores. They also give color. Okay, and these don't actually give color, but other chromophores are there. So these are, let's say they are not natural chromophores, but GFP and ETBR and M cherry, all of these are natural chromophores because they give color to the protein. But your tryptophan, tyrosine fluorophores, they are intrinsic fluorophores in proteins. For DNA, every nucleotide is a intrinsic fluorophore because they are having this ring structure. Okay, right. So now we will look at this. So we see that there is this xenon lamp. Xenon lamp is giving light. It is going to the mirror. Mirror is reflecting it. It is going to the monochromator. Monochromator selects one specific wavelength. Wavelength goes to the sample. From the sample, the light goes to another monochromator that is selecting the specific emission wavelength. Specific emission wavelength goes to detector, and then detector has photomultiplier tube, which is called PMT, and then from there it goes to data processing and output. Okay, that is the setup. It is called a fluorimeter. Now we'll talk about something called Stokes shift. What is Stokes shift? You already know this. Right now we talked about the fact that light is absorbed by the molecule and then light of a longer wavelength is released by the molecule. This difference in wavelength is only called Stokes shift. Okay, so lambda emission minus lambda excitation, that is Stokes shift. So fluorescence spectrometers typically consist of a light source, usually a high energy lamp or laser for excitation, for example xenon lamp, and monochromator to select specific excitation and emission wavelength, a sample holder and a detector to measure the emitted fluorescence. Okay, so biomolecular studies. Now these are the different applications for studying structure and dynamics of biomolecules such as proteins, nucleic acid, lipids, etc. and monitoring enzyme reactions, ligand binding, etc. And these are what we also normally do in the lab, okay, using fluorescence spectroscopy. Then there are different other techniques. For example, all of these spectroscopy based techniques that are there, visualizing cellular compartments and processing fluorescent labels, and then in vivo imaging of medical diagnostics and research, etc. All of these different things can be done, okay, using fluorescence spectroscopy. Now there is something called quantum yield. Now this often comes in exams. That is why I've included this. So what is this? Quantum yield is something very direct. So see, number of photons emitted divided by number of photons absorbed. If the quantum yield is 1 or 100%, that means that the particular sample, in that sample there is no loss of energy. So how many protons were absorbed, that many protons have been emitted, right? So quantum yield of 0 indicates no fluorescence. Okay, and there is something called Kr and there is something called KNR. So Kr emits light and KNR kills light without emission. So we'll see what is this Kr and KNR. Okay, these are actually probability terms. So see, Kr is actually radiative rate constant, and this represents the probability per unit time of radiative decay, that is emission of a photon. Okay, and KNR is not radiative rate constant. It represents all radiation less deactivation pathways such as internal conversion, vibrational relaxation, collisional quenching. So that means two molecules are like hitting against one another, some energy is lost, all of these different things are happening, right? So because of which all of these different things, there is some energy loss. So we are taking all of that into consideration as well, and that is your KNR term. So if the KNR is low and the KR is high, that means it is very strong fluorescence. It is a high quantum yield. If KR is low and KNR is high, that means there is very weak fluorescence or no fluorescence. Okay. Okay. Understandable until now. Now let's try to solve a few questions. Okay, so to record the fluorescence emission of lysozyme protein, lysozyme is an everyday protein that is used in the lab and in a protein biology lab. Okay, that is because lysozyme is used for breaking up cells, and it is a very stable protein which you can store, okay, for like a long period of time. So it is used to break open the bacterial cell wall and take out your desired biomolecules from there, mostly recombinant proteins. Okay, so this lysozyme protein has phenylalanine, tryptophan, tyrosine. One should select the excitation coefficient for which of the following. In the monochromatid, you have to make the setting, right? So whether you should set it for tryptophan or tyrosine or phenylalanine. This is a very practical question. Try to answer. Let's see if you guys know, and then I'll tell you the right answer. Okay, so tryptophan residue, since tyrosine and phenylalanine are non-fluorescent. Okay, so you're saying that it is C. So tryptophan residue since tyrosine. Acha. Yes, yes, that is correct. Good. That is very correct. So C is the right answer. That is because tryptophan has a very large absorption capacity compared to tyrosine and phenylalanine. Why is that so? Because of its indole ring largely conjugated pi electron system. Okay, because of that, mainly because of the structure, and also because tryptophan has very high Kr and lower Knr. That is because of which it has much better absorption capacity compared to phenylalanine and tyrosine. So if your protein by any chance has no tryptophan but some phenylalanine and some, let's say, tyrosine, then it's very difficult for you because absorbance would be very low for that protein. Okay, so that is a sad story, right? So now we will go to another question. The type of radiation that often triggers molecular vibrations. This is simple. Think about boiling water. What happens? You hear this sound, right? So what is happening? Why are you hearing that sound suddenly? Try to answer this question. It's something that you can answer, all of you. The type of radiation that triggers molecular vibration. A is microwave radiation and infrared radiation. Okay, let's see. Yes, that is correct. Very correct. So I was giving you the hint that when water boils, you get all this noise. Why? Because all these water molecules are colliding against the utensil, right? So, and that is because of vibrational transition, right? So because of which you are getting this kinetics, vibrational motion. Besides this, microwaves, they induce rotational transitions. Okay, but UV visible rays, they actually induce electronic transitions. And microwave induces molecular rotation and infrared causes molecular vibration. X-ray and UV are non-ionizing radiations. They possess higher frequency and higher energy, which is used for electronic excitation only. Okay. Right. Now we'll go to another question. Very simple question. An electron is excited from a non-bonding orbital to a pi orbital, which is anti-bonding. What is the correct representation? Non-bonding to pi bonding, anti-bonding orbital. C. Okay. Yes, correct. C is the right answer. Non-bonding is always represented by N. Pi bonding is by pi. And you have this star. The others were sigma. Right? So sigma is s orbital. It is not pi orbital. Right. So now we have some questions. Simple questions that you would be able to solve. Very simple questions. Now see, UV absorbance at 280 nanometer. Why 280? Because this is the wavelength at which tryptophan absorbs. Okay. So UV absorbance at 280 nanometer is primarily caused by tryptophan residues present in the protein. The absorbance of X molar protein in the experiment is 0.35. So A value is given to you as 0.35 at 280 nanometer, which is measured using a cell of 1 millimeter path length. The protein content in the cuvette approximately is something that you have to find out, and epsilon value at 280 of the protein is 10,000. So then you have A, you have L, you have epsilon. You only have to calculate C. C is the concentration. So using Beer-Lambert's law, you will calculate. Right? Try calculating. Point 0.35. Okay, let's see. Yes, that is correct. So 0.35 is the right answer. Why? Because D has to be 1 centimeter. It's given 1 millimeter here. So that becomes 0.1. Right? And what else? Baki, all the values are given. You have to just put them into the equation properly. And then we get the right value as 0.35 millimolar. Okay? Right. Now, spectrophotometers can be of two different types. Okay. One is single beam and the other one is double beam spectrophotometer. So we saw previously that there was this reference cell and sample, reference cuvette and sample cuvette, and that is called a double beam spectrophotometer. The one in which it just passes only through the sample, that is called the single beam spectrophotometer. The spectrophotometer helps in measuring the absorption of light by the sample. Now there is this question, okay? It's based on this double beam spectrophotometer. So this double beam spectrophotometer differs from a single beam spectrophotometer, since in the case of the former means double beam spectrophotometer, both sample and reference cuvette cannot be measured simultaneously. Reference cell lamp has different wavelength from single cell lamp. The chopper splits the light into two parts, which passes through the reference and sample cuvette. Reference absorption is first recorded, followed by sample absorption. So which one do you think is correct based on what you saw in the figure? So based on what you saw in the figure, which one do you think would be the right option? It should be C, but I don't know the correct explanation. Answer. Yes, it is C. So you see that there is this chopper which is actually separating the light out, and it is going to both reference and sample beam, and then it is coming to the monochromator, and then it is going into the detector. So why is that happening? Because we want to we want to basically know whether because of the sample present in the cell, what is the exact difference in you know about negative control, right? So negative control is important for every experiment, and here in the reference beam, what is there is the negative control. So the same light is going into both, and then this is giving some value, and let's say this is giving x value. So then the final value would be your x minus that sum value, right, to get the actual value. That is what is happening. And then the detector is actually subtracting it and giving to you. Okay, so this is actually negative control. So if you look at this, there is this light source and then monochromator, and it is going adjustable aperture, and then this is what is there in a single beam spectrophotometer. The entire light is going through the sample. There is no reference electrode. Okay, and there is this amplifier which is amplifying this and giving you the output. So this is the difference between the single beam and double beam spectrophotometer. Now we have another question on UV visible spectroscopy. So which of the following is true? They are asking you. An absorption band at 190 nanometer is due to disulfide bridge between two cysteine residues. The cuvette made up of quartz has two sides frost and two sides transparent. Now, a lot of you might have used cuvettes in your college days. I'm not sure in school days have you used? I never used in my school days anyway. So then some of you might know about cuvettes. Okay. And peptide bonds are chromophores with strong absorption bands at 280. Weak absorption band for peptide bonds come around 190, nanometer. Now from this question, you should understand that besides these aromatic residues, there are also other groups in proteins which absorb light. Okay, so it is recommended that you read about them, see which thing in a protein um, absorbs light at what wavelength. Okay, that is something that would be beneficial for these kind of MCQs. Okay, so we can try solving this. Let's see, any ideas, anyone? It's okay if you don't have any idea right now. So the right answer is actually B, because yes, B. B is the right answer. So cuvettes are having these. Cuvettes have two sides. Cuvettes are actually cuvettes, the quartz cuvettes. These are actually having two sides made up of frost and two sides transparent. Through the transparent sides, light travels, but the other two sides are relatively, you will see these are of some porous material, so it's for you to hold the cuvette there. Okay, and quartz is very expensive, and since light would never be passing through these other two sides, there is no quartz in those two sides. Okay, and it is also ensured that light is not lost from those two sides. So that is why it is frost. Now we will have why C is not the correct option. Achha, so why is C not the correct option? You can read about this. Okay, so peptide bonds, they are, whether they are chromophores, whether they are fluorophores, and what is their absorption and at which nanometer. You can read about this and you can tell me in the next class. Okay, so I'm not sure if these are chromophores. So you can read about this, and plus the bonds, like the wavelength. That wavelength you can check. What would be the wavelength of the peptide backbone? Okay, which like wavelength does it absorb light maximum? So then we can talk about that in the next class. Okay, I will also read it and then we'll see. Okay, which is the right option. Okay, so now we have another question. Monochromators are used to disperse white light into various wavelengths, which is true, they are asking you. At low concentration, light emitted from all parts of the fluorescence quartz cuvette can be considered almost uniform. Fluorophore and chromophore are the same thing. The maximum excitation wavelength for tryptophan, tyrosine and phenylalanine are 258 nanometer, 275 nanometer and 280 nanometer respectively. So which is correct according to you? A is correct. Achha. Let's see. Yeah, it should be. Yes, correct. So yeah, that is right. A is the right answer. Okay, the maximum excitation wavelength for tryptophan, tyrosine and phenylalanine, whatever is given here, that is exactly in reverse order. Okay, and fluorophores and chromophores, they are two different things, because chromophore gives color to that particular biomolecule. Fluorophore gives it fluorescent property. Okay, so if in the previous slide we saw that peptide bonds are chromophores, that would be wrong. And you can check the wavelength. I am not sure about the wavelength. I'll have to read about that too. Okay, but it is definitely not chromophore, because not all proteins are colored. Right? So only there is this RFP, GFP, YFP, etc. These are colored proteins, but not all proteins are colored. So then chromophore would not be the right answer in the previous one. And see, fluorophores are fluorescent chemical compounds that can re-emit light upon excitation, and chromophores are an atom or group whose presence is responsible for color of the compound. Okay, right. Now, the difference between a fluorophore and chromophore is given for your understanding. You can go through this. Okay, some of the examples are also given. Fluorescent label dyes are used for labeling DNA. There are different dyes, cyber, green, and all these different dyes are there. You can read about them. They often come in questions, and even in the upcoming classes, we'll talk a lot about different biophysical techniques, and there you will anyway come across a lot of different dyes. Okay, then we have heme group in hemoglobin for oxygen transport, which is giving hemoglobin this red color. Chlorophyll in photosynthesis, it is giving this green color to plants. Those are all chromophores, right? And absorption of light, so emits light, and okay, you can read about this and then we can go for the next question. Okay, so okay, now this amino acid absorbs in UV region. 280 nanometer is the wavelength at which it absorbs. You can already okay, so maximum lambda max is 280 mainly due to, so why do you think it absorbs at 280 nanometer? Because of which group do you think? Phenol group, correct. So all the aromatic amino acids, they are absorbing light very well because of these conjugated double bonds. Okay, and similarly all these nucleic acids, they are also absorbing light so well because of conjugated double bonds. Remember that. Okay, and what is this amino acid, by the way? Anyone? So absorption at 280 is mainly due to chromophoric moiety in the side chain. Here the phenolic group is the chromophore moiety. Okay. Now, which of the following statements is or are true? In fluorimetry, photovoltaic cell and PMT are used as detectors. PMT is photomultiplier tube. I was telling you about that. Right? When molecules are excited in the visible region, hydrogen deuterium lamp is used. When compounds have aliphatic carbonyl structure, they show more intense fluorescence than that of compounds having aromatic functional group. So which of these are true? And quantum yield quantifies the efficiency luminescence. Which are true? Efficiency of luminescence. So we can already see that some are wrong. B. Okay, let's see. Yes, that is correct. B is the right answer. So when molecules are excited in the visible region, tungsten lamp is used, not hydrogen deuterium lamp. Hydrogen deuterium lamp is for UV light. Okay. Now compounds having aromatic functional group, they show much more intense fluorescence compared to compounds having aliphatic carbonyl structure. Right? Okay. So for you to check is one of the things is that all these fluorophores that are there in proteins, are they chromophores too? So that is one of the things that you will check, and you will check the different functional groups in the proteins plus peptide bonds. What are they? Are they fluorophores? Do they absorb light at a particular wavelength, and which wavelength? Okay, so that is something that we can talk about in the next class after you guys find out. Acha. So now we have almost, this is our last question, right? So we have come to the end of the class almost. A researcher has prepared a new drug molecule and to study its interaction with a protein. So the lambda max of the drug molecule is at 260 nanometer. The desired emission wavelength should start from. So this person knows that the lambda max is 260 nanometers. So from where to where should its range be? So starting wavelength of emission should be what? Okay. Try answering. Maybe D. Let's see. And then we also have the explanation. Acha. So the explanation, acha. So the right answer is B. Why is that so? Because according to Stokes shift, actually we'll read this first. So the emission should start at least 10 to 15 nanometer ahead of the excitation spectra, since in Jablonski diagram the molecule once excited to S2 from S0, it comes down to the lowest vibrational level of S1. The energy gap from S1 to S0 is lower than that of S0 to S2. Hence, the fluorescence wavelength is higher than that of the excitation wavelength. However, much higher wavelength should be avoided since one may miss the emission spectra. Now, one important thing that you have to remember is for Stokes shift, you have to remember that it is often plus 10. Okay, so let's say this is your, so it is different for different molecules. Okay, it is different for different molecules, but the minimum is often plus 10. So if this is 260, then you should, based on here, if it's plus 10, it becomes like around 270 to more than that. So you should at least start somewhere around 270 for detecting the emission spectrum. Okay. So you can read more about this if there are any doubts. That's all for today's class. Right? Okay. So that's it. That's all for today's class. These are the different materials that I've used for making the PPT. And you can refer to these books that are there. Thank you for your patience. Thank you so much. If there are no more doubts, we can stop the class. We can end today's class. Yes, thank you so much. Thanks to all of you. Thank you for paying attention. Thank you for answering so many questions. Thank you for bearing with me on this. So we'll meet again next week, next Monday. Okay, bye-bye. I'm ending the call now.